Hi friends. In this video, we are going to talk about mutation, which is a change in the DNA sequence. As you can see here in the three DNA double helix, the one in the middle has this damage, right? It can be because of the mutation or by some chemicals. And we will learn about how to identify these mutants, like the mutants we have, what kind of mutant they are, and how to identify like whether the damage that has caused, like the mutation, is it spontaneous, like is it random, or it is induced by some chemicals. And to identify this, we will use different tests. So I will name some, which is replica plating test, Ames test, and fluctuation test. So please make sure to like, share, and subscribe my channel. So what is mutation? Mutation is a spontaneous change in the DNA sequence which can affect the protein, right? Like from the central dogma, the DNA can affect protein and like the RNA and the protein. And if the mutations are like a major changes, it sometimes can result in the genetic variation and possibly new characteristics or new traits. Because some of the mutations are silent mutations, which means that the mutation is there, but the protein remains the same. So the possibility of new traits is very less. But sometimes the mutation is like in the major region, which can change the protein, and we can see new traits. And it can occur like during DNA replication because if you know the enzymes that do the DNA replications, they have an error rate and they can cause sometimes during replications there are problems and it can like the mistakes can happen. It can be exposure to some mutagens like some chemicals in the environment or the DNA repair mechanism. As we know that when there is uh, some difference, like the correct base that we want is not placed, the DNA repair mechanism happens to correct the damage that like the mutation. But sometimes the error in this DNA repair mechanism can also cause mutation. And sometimes these mutations are harmful, but it can be advantageous as well. Like we will talk about the famous example, which is the sickle cell trait. So in sickle cell, there is mutation, like the one allele is fine, is normal, but the other one has the mutation. So it gives the trait the name the sickle cell because the red blood cells in that case are sickle. But the advantage of that is people with this sickle cell trait, they have less probability to get malaria as compared to the normal people. So if you go to regions like Africa where the malaria is very endemic, like it's very prevalent. So in that region, you will find lot of people with this mutation because it's advantageous that it protects them from malaria. So sometimes it can be advantageous, but then it can be harmful as well. And so there are different causes of the mutation. One is the natural region, which is like the UV radiation in and like different cosmic radiation, different type of radiation. It can induce genetic alteration. So this is the natural mutations that happens in the nature. It can be by some chemicals or pollutants that can also cause DNA damage. That's why when there are this radiation leakage in some areas, like in some area, like suppose if there is a chemical factory and there is leakage of some gas, so people from that area they suffer sometimes from different diseases. That's because that chemicals causes DNA damage. And then sometime, as we have talked about, mistakes happen during DNA replication and the repair mechanism, and that can also leads to DNA like to mutations in the DNA sequence. So now we know what causes the mutations and what are mutations. So there are different types of mutation that can happen, right? Uh, First is point mutation. Like there is only like if you know the protein is contains amino acids, and each amino acid is made of three bases from RNA, right? And sometimes from these three, there is only one base that can change. So a single base can result in a substitution of amino acid in a protein, right? So suppose there is a single change and the codon that is formed from three bases, it can change completely and form a new protein. And sometime it changes, but the mutations does not like have a significant effect because the protein remains the same. So sometimes it can be silent, in which the protein remains the same, there is not much change. But sometimes it can be tedious, like in case of sickle cell, it's the point mutation that happens. It can be some insertion or deletion, like during the repair mechanism or during the replication, some bases are either removed or inserted, which can lead to the frame shift, which means that suppose we have a sequence in which first three code for one amino acid, another three for another amino acid. But suppose you change the sequence and instead of starting from first base, we are starting from the second base. So now the codon is completely changed and now we have frame shift mutations which can be formed by either insertions and deletions, right? So, and the other is gene rearrangements. Like sometime when there is gene rearrangement during recombination, sometime there can be inversion, like the complete reverse of what we want, duplications or translocation. Like we have more instead of one copy, we have two copies of gene. So this can also cause mutations. And the another one is the loss or duplication, which is almost the same as the insertion, but in insertion or duplication deletions, there are specific bases that are inserted or deleted. But in duplication, the entire section is duplicated or lost completely during recombination. So these are major type of mutations that can happen in the nature, and this can be spontaneous or induced, like we have talked about. Like it can occur spontaneously, like from natural processes like the environmental factors or the DNA replication error, because this is something which is happening naturally and we cannot control these things. So this is spontaneous, like the example of spontaneous mutation. But there are some mutations that are induced chemically, like exposure to mutagen that we have talked about, like the leakage of some chemicals from some factory and the people around that area that they get affected, and this is the induced mutation. So it is very important to understand how the mutation occur, and this basically helps to identify the genetic variation and its impact on organism. Because if we know that the mutations is occurring spontaneously because of the natural processes, then this can help to identify like the evolution of the organism and how in future different mutations can play a role. So if it's advantageous and why the nature selected that. And if it is induced mutation, it helps to understand specific chemical behavior, like how they react and how they affect the uh, human organism, like in terms of diseases. So it's very important to understand why, like whether it's spontaneous or induced mutation. So this is what we are going to understand in this video. So we are going to identify these mutations by different mechanisms, by different tests. So first test is the replica plating test. So this test is basically to uh, so the basic principle is you replicate the bacterial population from one plate to the another. So basically the idea came from the PCR, like the replication of DNA using in the recombinant DNA research, right? So it derived from there, like if we can replicate this DNA sequence, why can't we replicate the bacteria from one plate to another? And when we know that, we identify like the mutants based on their growth, where they grow. So I will explain in detail. So what, as I have told you, it replicates on the petri dish. So we have a back like so I will give you an example. Like so it's just transfer the colonies from one plate to the another and then identify based on their growth and appearance, and they can be studied further to identify the genetic changes and their potential. So like here you can see what we do is, so it starts with the master plate. Suppose you have a plate in which you have all the all the there is no pressure actually. All the nutrients are present, or there is no antibiotic or anything, nothing is present in the master plate. So then you replicate this by transferring all the colonies on a velvet cloth, and then this velvet cloth you imprint the colonies on another plate of petri plate, right? So you have all the colonies on this petri plate, and then you grow them what you want to study. So you can grow them based on the antibiotics or based on the growth medium. Suppose you know that maybe in this plate some mutants can some colonies cannot, like they require a particular nutrient but the others not, but you don't know, so you can put specific medium, or you can like have a plate which lacks specific medium, and then you can identify the colonies. So when you grow them in different antibiotic plates, so this plate contain ampicillin and this plate does not contain ampicillin. So this plate, the plate in which there is no antibiotic, all the colonies will grow. But the plate which contains antibiotic, only the colonies which are resistant, like which can grow in the presence of antibiotic, will be present. So here you can see only out of five, three colonies grow, which means that these two colonies, they are ampicillin sensitive. So in this way, you can study these two colonies or the rest to identify what are the changes between these colonies, what are the changes in the sequence, and how can they affect the growth, and basically like if it's the media, how, if it's specific nutrient, how they change in the specificity of the nutrient. So this is one test to check the how the mutant grow in the presence or absence of the specific nutrient or specific antibiotics. So next test is the Ames test. Basically, it is a test to identify the substance which causes mutations. So it was basically developed by Bruce Ames, and that's why it's called the Ames test. So it is a bacterial assay to identify which substances causes mutation. So when this test was done, the Salmonella bacteria, it was used to identify like the exposes exposing this bacteria to the mutagen, like to some chemicals, and then observe, because this bacteria, they cannot, like they lack histidine, which means that they need histidine for their growth. If the histidine is not present, they cannot grow. So this test basically check in this setting, like if you put some chemicals, can you reverse this histidine, which means that because they cannot grow, because they don't have histidine, but is it possible that we put some chemicals and now these bacteria start growing and form colonies? So this test basically uses this, but there are other technique which can be used apart from salmonella. There are other bacteria which can be used, and then if a substance inducing more than twice the number of histidine reverting colonies, it is considered mutagenic. Like uh, if you see more colonies are happening which can grow, even the histidine you did not provide, which means that the substance is mutagen. So what they did is, so they have a strain of Salmonella typhimurium which is histidine negative, which means that they require histidine for their growth. So they grew that bacteria in this plate which lack histidine and but contain all the growth medium, like from the rat liver extract they use which lacks histidine. So when they don't contain anything, like not histidine, so they don't contain any chemical and lack histidine, nothing grows, because these strains require histidine for their growth. So when histidine is not present, they cannot grow. But then they place the chemical that they want to test in the wells and now plated these bacteria. So only if the chemical can cause more colonies, then only it will be considered mutagen. So then they incubate it. So suppose if a chemical causes only one colony, which can be random, so they do not consider this as mutagen. But here you can see most of the colonies grew from this histidine negative, which means that this chemical reverted the histidine negative. So now they all are histidine positive. So this chemical is considered to be mutagen, which reverses the histidine negative to histidine positive. So it finds a substance that causes mutation in a particular population of bacterial, like this is a bacterial assay. So Ames test is basically this. So now we are going to study the fluctuation test, which is also called the Luria-Delberg fluctuation test because it's developed by them. So basically, this test identifies the rate of spontaneous mutation. Like suppose you have a culture, if the mutation that we see is completely spontaneous, like it's random, or it is because of the some selective pressure. So this is basically to quantify the mutations. So basically what we need to do is to make different serial dilutions of the culture from a single colony and then study the mutation over generation. And by this researcher can estimate the mutations that happens per division, per replication, and then help them to study the evolution. So what happens is, so imagine like this is a flask which contains like tons of the E. coli, which is grown in the absence of T1 phages. So this is like you grow E. coli without the bacteriophage, and then when you grow them, you see this. So this ton R means so the bacteriophage, when they infect this the E. coli, they bind to a particular receptor to infect the E. coli cells, which is called the TON-B receptor. But when we make them mutant, like TON-R, the bacteria cannot, like the bacteriophage, it cannot infect, right? And it cannot bind to the receptor, and now they cannot infect, and the E. coli will grow. So here you can see we have tons of mixture from different colonies, and when you plate them, you will see that because they are resistant colonies, they will grow equally, right? But in this case, they all are, there is no mutation, right? I mean, they all are individual cultures and we did not plate them. They are only E. coli cells. And then we plate them in the presence of T1 phage. But because in this culture, they are all same culture, right? Individual cultures, but from a particular colony. In this case, because they all are like ton sensitive, then they are not ton R, right? So they cannot grow. Only the bacteria that acquired mutation randomly they will grow. And earlier they acquired mutation, the more they grow. So when we plate them, you can see the mutations. So the first culture, no colony, which means that everything is tone sensitive. They can, like the phage can infect them, and then the bacteria dies and they cannot form colonies. In the second one, you see only one colony. Like in the fourth one, you see like they begin to become resistant, right? But at later stages, because this is about multiple generation. But the only thing that you see is number 14 and 18. You will see tons of colonies, which means that the phages cannot infect them and then they replicate. And because there are so many in number, which means that the mutations occur at an early stage. Even when the phage was not present or something, it occurred at an early stage and that's why it grew too much, right? So which means that the mutations occur spontaneously and based on that you can see at which stage mutation occur, if it's at the early stage, if it's at the later stage, because here you can see they are at the later stage, but this one is very early. That's why you see so many colonies. So this is basically to test the random mutations and at which stage of life, like the division, they occur. So I hope it's clear, and if you have any doubt, please let me know in the comment section. Please like, share, and subscribe my channel.